So today we are going to discuss chapter number 1, cellular adaptations and cell injury. So here first I would like to introduce a concept of responses of this cell to various types of stimuli, right? So now we will start from this. What are the responses of these cells to various types of stimuli? So what I want to say you that cellular responses, right? So we are going to discuss about cellular responses to different type of stimuli. Cellular responses to the different type of stimuli, because these stimuli will decide whether cell will acquire adaptation or cell will go for injury, or there will be aging, or there will be intracellular accumulation of the substances like glycogen and protein and fat. So these are the 4 important changes which you will see in a cell whenever they are exposed to the stimuli. And remember here that stimuli type will be deciding what kind of changes we are going to see inside the cell. For example, one by one I will introduce you. When the stimuli is a altered physiologic stimuli, see, altered physiologic stimuli. So altered physiologic stimuli, when it is given to a cell, means the stimulus is not injurious and it will just change the physiology of the cell. So altered physiologic stimuli, or maybe non-lethal type of injurious stimuli, means that stimuli will be injurious but there will be no damage, or there will be no damage to the cell content. So altered physiologic stimuli or non-lethal stimuli So here it is very important to understand that whenever we are seeing this type of stimulus to a cell will have adaptation. So cell will acquire adaptation. So this will be giving rise to cellular adaptations. Right? Cellular adaptation. So cellular adaptation, cell will adapt themselves, right? So that they should not get damaged and they should maintain their normalcy when a stimulus is removed. So that is called cellular adaptation. So altered physiologic stimuli or non-lethal stimuli will create cellular adaptation. So what are the examples of the cellular adaptations? So examples of the cellular adaptations I am writing here, and later on we will discuss about them, right? So what are the examples of the cellular adaptations? Number 1, examples of the cellular adaptation. Number 1 is atrophy, hypertrophy, hyperplasia, and metaplasia, right? So these are 4 types of cellular adaptations which examiner asks in exam like this. What is hypertrophy? Question was, what is hypertrophy? In All India exam, question was, what is hypertrophy? It's a cellular adaptations, right? So similarly, examiner can ask many other questions like, what is atrophy? What is hyperplasia? What is metaplasia? So all these are examples of the cellular adaptations. Cellular adaptations are the responses to those types of stimuli which are physiological, within physiological limit, or which are having non-lethal nature. So that is how we will understand what is the meaning of cellular adaptation. Means whenever altered physiologic stimulus is given or non-lethal stimuli is given, we will see cellular adaptations, and these are 4 important types of cellular adaptations, right? Later on we will discuss detail about cellular adaptation. So I was talking about the responses, various types of cellular responses to the stimuli. So second type of stimuli, now we are going to see that second type of stimuli which we are going to see will be a lethal stimuli. See, in cellular adaptation it was non-lethal stimuli or altered physiological stimuli. Now we are going to see what will happen when there is a lethal stimuli. When lethal stimuli is given to a cell, what are the changes you will predict now? Lethal stimuli, right? So now it depends on what is the duration of the lethal stimuli. Was it transient or was it persistent, right? So these are two types of lethal type of stimuli. So when we see this stimuli is transient, when this lethal stimuli is transient, this will give rise to reversible injury. So here starts cell injury, reversible cell injury, right? So reversible cell injury will be given when this lethal stimuli will be having transient period, right? But when this lethal stimuli is persistent, what will happen? What will happen? So persistent stimuli will be causing irreversible injury. Irreversible injury, right? So persistent stimuli will be giving rise to irreversible injury. So transient stimuli, reversible injury, persistent lethal injury stimuli will be giving irreversible injury, and we all know that after irreversible injury, what will be the fate of a cell? What will happen? Cell will be dead, cell death, right? So after this persistent stimulus, after this irreversible damage, what we will notice that there is a cell death. And after cell death, there are morphological changes. What are those morphological changes? So what are those morphological changes? We have to understand. So after cell death, there will be morphological changes. And these morphological changes are necrosis, apoptosis, right? Necroptosis and pyroptosis. So that is how we can understand what are the changes we are going to see in a cell to a different type of stimuli. Right now we were talking about the lethal stimuli. When it was transient, we have seen it was a reversible. When it was persistent, irreversible injury. After that, cell will be having death, and after cell death, they will be having necrosis, apoptosis, necroptosis, pyroptosis. All these are the morphological spectrum after cell is dead, right? So these are the important things about lethal stimuli. Now there is a third type of response. What is the third type of response? Sometimes that injury is chronic, right? And because of this chronic injurious stimuli, cell will develop another type of change. And that another change is they will acquire the substances in their cytoplasm. In simple words, intracellular accumulations. Because of the chronic stimuli. So now we will see that because of the, so third one, because of the chronic injury or chronic injurious stimuli. Because of this chronic injurious stimuli, what will happen? When chronic injurious stimuli is given, there will be intracellular accumulation. As I said, within the cell, intracellular accumulations. So within the cell, you will find intracellular accumulations. What are these intracellular substances? So when there is a chronic stimulus given to a cell and they are causing injury, they can have accumulation of fat, glycogen, protein, right? So these are the substances which may be accumulated inside cytoplasm, like fat, glycogen, proteins, right? So these are the various substances which may accumulate. Along with this, we have to understand this may be a reason for calcification also, right? So this may be the reason for calcification also, because calcium will be deposited within the tissue or within a cell. That is also because of the chronic injurious stimuli. So now we can see that how we started. When the stimulus was altered physiologic stimuli or sublethal stimuli, cells were undergoing adaptations. When it was lethal, cells were having a reversible injury when it was transient, and when it was persistent, causing cell death because of the irreversible injury. And after that, we were seeing that necrosis, pyroptosis, all those morphological changes will be happening inside the cell. And when this stimuli is chronic injurious stimuli, within the cell, there will be accumulation of certain substances. What are those substances? These are fat, These are glycogen, can be protein, and can be the calcium also. So that is what we are calling them, calcification. So this is the third type of response of a cell to the stimuli. Fourth type of stimulus, another important stimulus which is very, very important for us to understand human physiology, is the fourth one. When there is a sublethal, see that what we have read, there was non-lethal first, then we have read lethal stimuli, then now we are seeing the sublethal, means it is not the injurious or not the causing cell death, but that just below that lethal level, sublethal stimuli. And this sublethal stimuli will be persistent. So this sublethal stimuli is persistent. So persistent sublethal stimuli, what it will cause? Because of the persistent sublethal stimuli, there will be accumulation of the damaged protein, damaged structures, and organelles will be damaged, or there will be some changes in the molecular structure of the cells. In simple words, they will induce aging. In simple words, they will induce aging. Persistent sublethal stimuli to a cell will induce cellular aging. So this will induce cellular aging. So these are the four important cellular responses to different type of stimuli. Now we will see one by one from the first one, altered physiological stimuli. Now we will see what I had explained you. When there was altered physiologic stimuli, there were cellular adaptations. What are the cellular adaptations? Atrophy, hypertrophy, hyperplasia, and metaplasia. So now we are going to discuss one by one. So number one topic we are going to discuss is the cellular adaptations, right? So number one is cellular adaptations, right? So we have seen a brief discussion about when cellular adaptation will happen, when there is a altered physiological stimuli, or when there is a non-lethal stimuli given to a cell, right? So the question is, what is cellular adaptation? What is cellular adaptation? Cellular adaptation, when this stimuli was given, already we have discussed that stimuli is given to a cell, right? And we know that stimuli was altered physiological stimuli or non-lethal stimuli. So when a stimulus is given to a cell, what will happen to the cell? Cell will be having change in the number. Cell will be having change in their number, size, phenotype, and functions. So all these things will be changed, right? So because of the stimuli, all these things will be changed. So when altered physiological stimuli is given to a cell, there will be change in number, size, phenotype, and functions of the cell. And once you remove the stimuli, so after removal of the stimuli, cell will come back to the normal state. Cell will come back to the normal state. So that is the important point to notice here. What is cellular adaptation? When a stimulus was given, there was a change in their number, size, phenotype, and functions. But when you remove the stimuli, they will come back to the normal state. So what does that mean? Cellular adaptations are completely reversible. That was the question in All India exam also. So we should understand these cellular adaptations are reversible conditions, right? So cellular adaptations are reversible. Cellular adaptations are reversible. That is important point we have to keep in mind. You remove the stimuli, they will come back to the normal state. That is the meaning, right? So that is the meaning, and that is only because of the altered physiological stimuli or non-lethal stimuli. That is why they are reversible. Now there is another question comes to our mind. These cellular adaptations are physiologic or pathologic? Physiologic or pathologic or both? So answer is simple. They are both physiologic and pathologic. So this point is also very important to understand. Cellular adaptations are both physiologic. So cellular adaptations are both physiologic and pathologic, right? So they are both physiologic and pathologic. So this is very important to understand कि cellular adaptations are both physiologic and pathologic. For example, if you talk about the physiologic, we can take example of the pregnant uterus. So what are the changes we are seeing in pregnant uterus? Hypertrophy, hyperplasia. So that is the example of physiologic cellular adaptation. And pathologic example, I will give you example of the Barrett's esophagus, right? Barrett's esophagus. We will discuss about Barrett's esophagus later, right? But just for the sake of understanding, what I wanted to tell you, these two important things. Cellular adaptations are completely reversible, number one, and they can be both physiologic and pathologic. That is also very important to understand. Cellular adaptations are both physiologic and pathologic. This is very important to understand if examiner asks in your future exam. So that time we should remember that cellular adaptations are both physiological and pathological, right? And I had given you example. So one by one we will see what are the examples of the cellular adaptation. So number one example which we are going to discuss in cellular adaptation is hypertrophy. So number one example in cellular adaptation is hypertrophy. What do you mean by hypertrophy? Whenever there is an increase in size of the cell, and because of increase in size of the cell, overall size of organ will be also increased. So in hypertrophy, what we need to remember, what is the definition of the hypertrophy? Definition of the hypertrophy is size of the cell will be increased. So in hypertrophy, size will be increased, but number of the cells will remain same. So numbers will remain same. So cell number will be same, but cell size will be increased. That is the hypertrophy. That is the meaning of hypertrophy, right? But why? Why this hypertrophy happens? What is the mechanism? Now the question is, what is the mechanism of hypertrophy? So the second thing, what is the mechanism of hypertrophy? Mechanism of hypertrophy is increased in synthesis of cellular protein. So increased cellular protein synthesis. So because of increased cellular protein synthesis, there will be increase in the size of the cell, and that is called hypertrophy. So mechanism is increased in cellular protein. Now another question comes to our mind, just like if you remember what I said in cellular adaptation, they are both physiologic and pathologic. Now the question is, what is hypertrophy? Physiologic, pathologic, or both? So answer is both, same like other cellular adaptations. So hypertrophy is both, remember it is both physiologic, as well as pathologic, right? So hypertrophy is physiologic also and hypertrophy is pathologic also. Physiologic and pathologic. What is the example of physiologic hypertrophy? Physiologic hypertrophy example will be simple example, pregnant breast or pregnant uterus, not breast, pregnant uterus, right? So physiologic example is the pregnant uterus. So during pregnancy uterus size will be increased, and that is example of physiologic hypertrophy. What I want to convey you a simple message that pregnant uterus, what you are seeing is example of both hypertrophy and hyperplasia. Both hypertrophy and hyperplasia. Both hypertrophy and hyperplasia. So that is important to understand because this is exam question. Examiner asks you, pregnant uterus is an example of, that time we must remember that it is both hypertrophy and hyperplasia. Remember, it is both hypertrophy and hyperplasia. So that is important MCQ in exam, right? So this is very important to understand. The pregnant uterus is an example of both hypertrophy and hyperplasia, right? What is the pathologic hypertrophy? Pathologic hypertrophy we often see in cardiomegaly because of the valvular regurgitation, right? Because of the falling back of the blood flow into the ventricle. Because of the consistent pressure on the ventricle, there will be stretching and hypertrophy, right? So that is called pathologic hypertrophy. So it is cardiac enlargement. So cardiac enlargement is an example of the pathological hypertrophy, and this cardiac enlargement is due to valvular defect. Due to valvular defect. Right? So these are examples of the physiologic and pathologic, just to make you understand what is the exam question and to understand that hypertrophy is both physiologic and pathologic, and these are examples, right? Now coming to the third one, coming to the second one, sorry. Second cellular adaptation. Second cellular adaptation we are going to discuss here is the hyperplasia. Hyperplasia. What do you mean by hyperplasia? So again, what is the definition of the hyperplasia? What is the definition of the hyperplasia? So you can see the name itself is telling there is an increase in number of cells. Plasia means number. So number of cells will be increased, right? You remember, number of cells were constant in case of hypertrophy, but number of cells are increased in case of hyperplasia, and size will remain same. Size will be same. So size of cell will be same, but number of cells will be increased. That is known as hyperplasia. Number is increased. Why? Why number is increased? What is the mechanism? So here we will see what is the mechanism of hyperplasia. What is the mechanism of hyperplasia? Mechanism of hyperplasia is because of increase in growth factor, right? So whenever requirement of cell is more, growth factor will be more, and there will be more nuclear transcription factor, there will be more cellular proliferation. In simple words, I would like to tell you, whenever there is an increase in growth factor, because of the demand, right? Because of the demand of whom? A cell. A cell is demanding. See here, I am showing you, this is the cell and this is the nucleus. This is the nucleus of the cell. Demand of the cell was high, so that is why growth factor was increased. What will happen when there is an increase in growth factor? This growth factor will be sending signal to the nucleus. When nucleus will be having signal, nucleus will be synthesizing increased amount of nuclear transcription factor. Nuclear transcription factor. And because of synthesis of increased nuclear transcription factor, they will synthesize more proteins, and that more proteins will help in more cellular proliferation. So this nuclear transcription factor, in simple words, which I want to tell you, they will help in increased cell proliferation. So this is the simple basic mechanism of the hyperplasia. Why there is a hyperplasia? Because of increase in growth factor. Increased growth factor will promote more synthesis of nuclear transcription factor. This will allow more cellular proliferation. In simple words, in a very simplified manner, we can understand this increase in growth factor is the most important mechanism. Remember, for All India exam point of view, this is the most important mechanism for hyperplasia. Increased growth factor. Increased growth factor is the most important mechanism for hyperplasia and could be a future question also, right? So this is the number one. This is the number one mechanism. There is another mechanism, and another mechanism is also holding the same thing. Another mechanism is also holding the same thing. Here, there will be increased synthesis of cells from the tissue stem cells, right? So there will be increase in number of tissue stem cells, and from these tissue stem cells there will be increased synthesis of the cell. That is the simple. This is the another important mechanism, right? So hyperplasia because of the increase in growth factor, and second thing is because of increase in the tissue stem cell, which is synthesizing more number of cells. So that is the another important and less dominant mechanism. Less. What is the most dominant or most important mechanism? Growth factor. Remember, for future exam point of view, when examiner will ask you what is the most important factor, you must remember growth factor is the most important factor for hyperplasia. So this can be our future question also in All India exam, right? Now the same thing as we have discussed in cellular adaptation beginning, cellular adaptations are physiologic and pathologic both. So same way hyperplasia is also both physiologic and pathologic, right? So here again I will tell you hyperplasia is also both physiologic and pathologic. So hyperplasia is physiologic and pathologic. So both physiologic and pathologic. So here also we can see physiologic and pathologic hyperplasia. What is example of physiologic hyperplasia? Already we have discussed pregnant uterus, right? So again I am writing here so that we should understand, because it was already a question also. So pregnant uterus, and during pregnancy breast tissue. This is also one example of the physiologic hyperplasia. So pregnancy, uterus during pregnancy, breast tissue during pregnancy, they are example of the physiologic hyperplasia. Demand will be more hormone will be released, and these growth factor and hormones will induce more cellular proliferation. Hyperplasia will happen. That is the physiologic hyperplasia. Pathologic hyperplasia. What is example of pathologic hyperplasia? When there is a excessive stimuli from the hormones, right? Whenever there is a excessive stimuli from the hormones, for example, there is a increased estrogen. Because of increased estrogen on the endometrial tissue in uterus, on the endometrium of the uterus. So because of the increased estrogenic hormone stimulation, so this estrogen will be hyper, right? So they will be increased because of the excessive estrogen stimulation given to the endometrium. This will cause endometrial hyperplasia. This will cause endometrial hyperplasia. So this endometrial hyperplasia is example of pathologic hyperplasia, right? So now we can see these two examples of the physiologic and pathologic hyperplasia. What we need to understand here, there is one more thing that is called compensatory hyperplasia. Compensatory means they are doing compensation. For example, when we remove a part of liver regenerate themselves up to certain extent, and that is example of compensatory hyperplasia. So this is example of the physiologic hyperplasia. So in physiologic hyperplasia category, I would like to mention another important thing which is called as compensatory hyperplasia. Compensatory hyperplasia. What is compensatory hyperplasia? Compensation for the loss of tissue. So you have removed the tissue for the transplantation purpose, and to overcome that loss of tissue, there will be regeneration of the remaining tissue from the remaining viable liver tissue. So that is called as compensatory hyperplasia. So compensatory hyperplasia, what is example? Liver regeneration, right? So liver parenchyma cells will proliferate, and that is another example of the compensatory hyperplasia. Liver regeneration after transaction of the liver, or after the liver transaction, or after liver resection. So whenever we are doing liver resection for the transplantation purpose or for some pathology, there will be liver regeneration, and that liver regeneration will be example of compensatory hyperplasia. And this was the All India question in 2015. Liver regeneration is an example of hyperplasia, right? So that point we have to keep in mind. Liver regeneration is an example of hyperplasia. What type of hyperplasia? Compensatory hyperplasia. So that was the question in All India exam, right? So this is about hyperplasia. Now we are going to see the third one. Third cellular adaptation, third cellular adaptation is atrophy. Third cellular adaptation we are going to discuss is atrophy. Another important topic. This topic is very important because many questions had been asked from this topic, atrophy. What is atrophy? What is the definition? So when you discuss the definition of the atrophy says size and number of cells both will be decreased. Size, number, both will be decreased. So that is interesting to understand that during atrophy, size and number both will be decreased. So during atrophy, what I am saying, size and number both will be decreased, and because of that, organ size is also reduced, right? Now, what is the mechanism of the atrophy? That is interesting, and that is the question which examiner is very fond of in recent exams. We have seen they are asking many questions from this topic called atrophy, right? So just mark it. This topic is very, very important for all India exam. Atrophy. So what is the mechanism of atrophy? What is the mechanism of atrophy? Atrophy, basically there are three important mechanisms, right? So here I will tell you, in mechanism there are three important mechanisms, right? Number one is decreased in protein synthesis. Simple. Protein synthesis is gone down, and that is why atrophy will happen, right? Simple. So number one is decrease in protein synthesis. Number two and number three. Number two and number three, sort of, I would like to tell you, they are interconnected to each other. See this arrow. Why I am writing? They are interconnected, means they are interrelated to each other. What are these two other mechanisms which examiner ask in exam? This mechanism is, number two is ubiquitin proteasome degradation pathway. Ubiquitin proteasome degradation pathway. So ubiquitin proteasome degradation pathway, this is another important thing which examiner had asked in recently DNB exam as well as All India exam. So ubiquitin proteasome degradation pathway is associated with atrophy, and the third thing is the autophagy. Autophagy, and these both are interconnected to each other. That is what I have shown by this mark, right? So ubiquitin proteasome degradation pathway is very, very important. Whenever you read this in any subject, you must focus on this topic, because in All India exam they are asking question from here, right? And autophagy was another important question which examiner had asked in All India exam 2014 and 15, right? So that is why I wanted to discuss about these three mechanisms of the atrophy. If you look at the three mechanisms, what I have written, decrease in protein synthesis, so it is very clear, given protein synthesis will be gone down, so obviously cell size and number will be reduced. Now coming to these ubiquitin proteasome degradation pathway and autophagy. First we should understand what is the meaning of autophagy. Always keep that thing in your mind. Some of the medical terminology, the name itself will be revealing about themselves. For example, what is the meaning of autophagy? Question is, what is the meaning of autophagy? Auto means what? Auto means self. Auto means self, and phagy means eating, right? So self-eating is autophagy, right? Self-eating is autophagy. So that is very important thing to understand. This autophagy is one of the mechanisms responsible for the atrophy. Decreased protein synthesis, ubiquitin proteasome degradation pathway, and autophagy. I said both are interconnected to each other. How they are interconnected to each other? These two pathways are intracellular protein degradation pathway. So here the concept starts. What are these two and how these two pathways are interconnected to each other? So what I want to tell you in the beginning, these both are collectively known as intracellular protein degradation pathway. Degradation pathway. So that is very important to understand. Intracellular, within this cell, protein degradation will happen, and that is the reason for atrophy. So how this protein will be, degraded within the cell, and how they are interconnected to each other. What is the connecting point? Connecting point is the ubiquitin. Connecting point is ubiquitin. So right now I would like to tell you, when these intracellular protein degradation pathway is activated, number one, you will see there is a activation of an enzyme. This is called as ubiquitin ligase. Ubiquitin ligase is activated. When ubiquitin ligase is activated, I am talking about, remember, I am talking about both ubiquitin proteasome degradation pathway and autophagy, right? So both pathways are having the same common thing, that is the ubiquitin ligase activation. Because of ubiquitin ligase activation, both will be having release of ubiquitin protein. When ubiquitin protein, so that is why I said the common thing to both of them is the ubiquitin. Because of the ubiquitin ligase activation, ubiquitin has been released, and this ubiquitin has a tendency to combine with the target protein, which has to be degraded, right? So this ubiquitin will combine with target protein. So this will combine with target protein, right? So this will combine with target protein. So that is the important thing to understand. Okay, this is the common pathway. This is the common thing which is seen in both autophagy and ubiquitin proteasome degradation pathway. So ubiquitin, when they will combine with target protein, what will happen next? After this, there are two possibilities. This will activate proteasomes. What are the proteasomes? These are a special type of organelle. Remember, these are a special type of organelle which will degrade this target protein which has combined with ubiquitin, right? So this proteasome will cause degradation of ubiquitin plus target protein. So they will be causing degradation of ubiquitin and target protein. So this is ubiquitin proteasome degradation pathway. That is why it is called as ubiquitin proteasome degradation pathway. So proteasome is a special type of organelle which will be activated when ubiquitin and target protein has been combined, and this will cause ubiquitin proteasome degradation. So this is how we can see ubiquitin proteasome degradation will happen, and protein will be degraded, atrophy will happen. So this is the reason for atrophy, right? So because of this, there will be atrophy, right? So because of this, there will be atrophy, right? So because of ubiquitin proteasome degradation, there will be release of ubiquitin which will combine target protein. Proteasome will be activated, and organelle which are specialized for degradation of the protein which is been attached to ubiquitin, right? So this is the ubiquitin proteasome degradation pathway. Now let us see what happens in autophagy. Autophagy means self eating. So in autophagy, what happens after this target protein is combining with the ubiquitin? They will be taken into double membrane bound. See what I'm writing? Double membrane bound. This is called as autophagosome. So this is how autophagosome will be formed. Autophagosome. So autophagosome is what? It's a double membrane structure. See what I'm writing here? It's a double membrane structure. So this double membrane structure is autophagosome, and what it will contain? So this double membrane autophagosome has been formed, and this will be containing ubiquitin plus target protein, right? This will be containing ubiquitin plus target protein. What will be the next thing? Next thing, this will combine with lysosome. So now I am showing you, this is the lysosome, right? So this will combine with lysosome. So what I'm showing you, autophagosome is combining with the lysosome, and now lysosomal enzymes will induce degradation of the protein. degradation of proteins. So that is how degradation of proteins will be performed in lysosome. And that was the question in All India exam 2016. Autophagy is done by which organelle? Autophagy is done by which organelle? Lysosomes or mitochondria? Answer is lysosome. So that was All India question. So lysosome is the right answer. Autophagy happens within the lysosome. So now we can see that three important mechanism and basic essence of all of them is the intracellular protein degradation, right? So decrease in protein synthesis, second is ubiquitin proteasome degradation, third is autophagy. So all are associated with atrophy, right? So this is the mechanism of atrophy which we have discussed here. Same as we have discussed in hypertrophy and hyperplasia, they are both pathologic and physiologic. Same way, atrophy is also both physiologic and pathologic. Physiologic and pathologic. So both physiologic and pathologic. Atrophy is also both physiologic and pathologic. So what are examples of the physiologic atrophy? What are the example of physiologic atrophy? Physiologic atrophy example is the fetal development. During fetal development, there will be loss of notochord and thyroglossal duct. So thyroglossal duct and notochord will be present during fetal development, but due to atrophy this will be slowly atrophied during the development process of the embryogenesis and they will not appear, right? So during fetal development, loss of or atrophy of notochord, right? So atrophy of notochord and thyroglossal duct. So these are two things, right? So during embryogenesis or during fetal development, loss of notochord and thyroglossal duct is example of the physiologic atrophy. This is physiologic atrophy. What is pathologic atrophy? Pathologic atrophy, you will notice during the denervation atrophy. Denervation atrophy. What happens in denervation atrophy? Suppose there is a muscle, right? This is the muscle, and every muscle we know that they are having nerve supply. Muscle is having nerve supply, right? This is the nerve supply, right? So sometime because of the trauma or some injuries, this nerve supply will be lost, right? So what I said, because of the trauma or injury. So because of the some trauma or injury, nerve supply will be lost, and as a result of this trauma, when nerve supply is lost, muscle will be having atrophy. So this muscle atrophy due to loss of nerve supply is called as denervation atrophy, right? And this is another example of pathologic atrophy, right? So these are important things about the atrophy. Now I want to discuss some important things which examiner had asked recently in exam. Question is endometrial adenocarcinoma. See what I am going to discuss now. This is the important MCQ which examiner had asked in All India exam 2014 and 16, and this was repeated in JIPMER exam also recently. So it's very important to understand कि this kind of question is a proper example to understand कि during exam time you should read the question very carefully, and after reading the question, when you are giving your answer, that time also you should see all the responses very carefully and attentively, A, B, C, D, and then you give your answer. Don't become prompt or impulsive for giving these answers. For example, question was very simple: endometrial adenocarcinoma. Endometrial adenocarcinoma. Endometrial adenocarcinoma is associated with, this was the question in All India exam, right? Endometrial adenocarcinoma is associated with, option number 1 was hyperplasia. Option number 2 was atrophy. right? So when this question came in exam, many of the students, they thought it is hyperplasia. Endometrial hyperplasia is causing endometrial adenocarcinoma. So they gave answer only A. But that is what I wanted to tell you. When examiner asks you such kind of MCQ, read the options carefully and fully. A, B, C, D. Because there was option C where it was written both of the above. And that was the right answer. Both of the above is the right answer. Right? So remember that endometrial adenocarcinoma is associated with both hyperplasia and atrophy. So atrophy is also associated with endometrial adenocarcinoma. That was the question in JIPMER exam, right? So now we have to understand what kind of cancer. What kind of cancer is because of hyperplasia? Hyperplasia will be giving rise to type 1 endometrial adenocarcinoma. Hyperplasia is associated with type 1 endometrial adenocarcinoma and atrophy is associated with type 2 endometrial adenocarcinoma. This is how you have to think about the future MCQ. So this can be future MCQ. Ki hyperplasia of the endometrium is associated with, future question will be type 1 endometrial adenocarcinoma. Future question can be atrophy is associated with type 2 endometrial adenocarcinoma, right? So that is how we have to keep on studying. We have to keep on thinking what kind of question examiner can ask in future exam. And this is one perfect example which examiner had asked in both All India and JIPMER exam, right? So hyperplasia, atrophy, both of the above is responsible for endometrial adenocarcinoma. And another future question which examiner can ask you from the Robbins itself, atrophy is associated with poorer prognosis in comparison to the hyperplasia. So this point is also very important to understand. Type 2 is having poorer prognosis. So atrophy is having poorer prognosis, whereas type 1 will be having good prognosis. So that is also very important. And that is how we are going to predict the future question. So keep on predicting future question while you are studying any topic. That will be the essence of cracking exam with a good rank, right? So this point we have to keep in mind, that endometrial adenocarcinoma is both because of hyperplasia and atrophy. So hyperplasia, atrophy, both are precancerous. What I said? Both are precancerous, and that was PGI question, and that was the PGI question. So that is what I wanted to tell you. Both are precancerous lesion. Both are precancerous lesion, and this was the PGI question. Precancerous lesions, right? So now we have to understand precancerous lesions are hyperplasia and atrophy, right? So these are the important point about atrophy. Now we are going to discuss the last cellular adaptation, which is metaplasia, right? So now we are going to discuss the last one is the metaplasia. So fourth cellular adaptation is metaplasia. What is metaplasia? This is little unique topic to be discussed, right? Metaplasia is a, if you see the definition of the metaplasia is one mature cell has been replaced by another type of mature cell. So one type of mature cell is replaced by another type of mature cell, So one type of cell is replaced by another type of mature cell is called as metaplasia, right? So one type of mature cell is replaced by another type of cell is called as metaplasia. So here we have to be very careful, because this topic is also a very interesting topic and very frequently asked question in our exam, especially All India, AIMS and JIPMER exam, right? So definition is the one adult cell has been replaced by another type of adult cell or another type of mature cell. But the question had been asked in both All India and JIPMER exam, ki what is the basic mechanism of metaplasia? What is the mechanism of metaplasia? What is the mechanism of metaplasia? Is it the mature cell reprogramming or is it the stem cell reprogramming? That was the beautiful question. So remember, it is not the mature cell. Definition is mature cell has been replaced by another type of mature cell, but the logic, but the concept is stem cell reprogramming. How? Because of cytokine release during inflammation or during any other process, or because of growth factors. So because of the cytokine or growth factors which are produced in our body due to some of the non-lethal stimuli, what will happen? This will alter the tissue stem cell. So this will alter the tissue stem cell. And because of this alteration in tissue stem cell, there will be reprogramming. So it is because of tissue stem cell reprogramming. So that is the answer in All India and JIPMER exam. Tissue stem cell reprogramming is the basic concept of the metaplasia. So remember, definition and mechanism of metaplasia both are entirely different. Definition is one mature cell has been replaced by another type of mature cell, but concept or mechanism is still tissue stem cell reprogramming, and that was the answer in JIPMER as well as All India exam. So it is stem cell reprogramming which is responsible for the metaplasia, right? Now we'll discuss about metaplasia. What are the types of metaplasia? So first I will tell you, metaplasia can be physiologic also, can be pathologic also. So that is very important, because in the beginning itself we started like this, ki cellular adaptations are both physiologic and pathologic. So here again I will tell you, same way metaplasia is also both physiologic and pathologic, right? So both physiologic and pathologic. So metaplasia is also both physiologic and pathologic. So what is physiologic example? What is physiologic example? Physiologic example you will see in cervix. In cervix during menstruation, right? So during menstruation, what you are going to observe that there is a squamous metaplasia at the squamo-columnar junction of the cervix, right? So remember this point, because examiner may ask this question, because these are the simple things which we don't often see in our standard books, right? So this kind of question examiner can ask, do you know any example of the physiologic metaplasia? Answer is yes, I know, cervix. What kind of metaplasia you will see in cervix? It's a squamous metaplasia. So remember, it's a squamous metaplasia. Remember, it's a squamous metaplasia which you will see in cervix at a squamo-columnar junction during the menstruation, because of the hormonal changes, the squamous cells will be extending towards the columnar junctions, right? And that is the reason, that is the important example of the physiologic metaplasia. Pathologic metaplasia, there are numerous. So pathologic metaplasia, again I would like to tell you, there are two types of pathologic metaplasia. One can be epithelial and second is the connective tissue or mesenchymal, right? So two types of metaplasia I am going to discuss here. Epithelial metaplasia, right? So pathologic, one is epithelial metaplasia and second will be connective tissue metaplasia. Connective tissue metaplasia, right? So these are two types of metaplasia, epithelial metaplasia and connective tissue metaplasia, right? Epithelial metaplasia and connective tissue metaplasia I would like to discuss earlier because it's a small one. So what is example of connective tissue metaplasia which examiner may ask you in exam? Example is the myositis ossificans. See, the name itself is telling myositis ossificans. What does that mean? Whenever there is a trauma, right? After trauma, there is a muscle injury, myositis, right? There is a muscle injury. So in this muscle, after the muscle injury, ossificans, you can see after that injury of the muscles, blood vessels will release certain growth factor which will help in formation of the bones, right? Bones within the muscles after the trauma is myositis ossificans. Bones within the muscle. So after muscle injury, there will be formation of the bone. So there will be bone formation, right? There will be bone formation. So this is example of connective tissue metaplasia. After the trauma, muscles will be injured, blood vessel will form the new bone, and this is called as myositis ossificans. So always keep that in mind. Okay, myositis ossificans is an example of connective tissue metaplasia or mesenchymal metaplasia. This is also called as mesenchymal metaplasia. Mesenchymal metaplasia. So this is very important to understand. Mesenchymal metaplasia, epithelial metaplasia, both type of metaplasia can be seen. What is the epithelial metaplasia and what is the most common epithelial metaplasia in human body? That is another important and interesting fact which examiner can ask you. Epithelial metaplasia, what is the most common type of epithelial metaplasia? So this can be a important future question. Most common type of epithelial metaplasia, I will give you example. Example is the respiratory tract of the smoker. When somebody smokes, right, what are the changes you are going to see in a respiratory tract? What is the lining of respiratory tract? See, I am giving you example of most common type. In respiratory tract, you will often see there is a columnar cells, column-like tall cells, right? So I am showing you columnar cells. Respiratory tract is having columnar cells, right? And because of this smoking, right? So because of this smoking, there will be a stress, right? So because of this smoking, there will be stress, right? And this smoking will stimulate tissue stem cell. Mind my language. What I said. Tissue stem cell, not the mature cell, not the mature columnar cell. They will stimulate tissue stem cell, and this tissue stem cell will cause reprogramming, and as a result they will be forming a squarish flat squamous cells. So now squamous cells are formed. So these are squamous cells, right? So these are squamous cells which are formed. So now we can see the columnar cells have been changed into squamous cell. And what is the nomenclature of this metaplasia? What is the nomenclature of this metaplasia? So always remember, the end outcome of the metaplasia should be the nomenclature of the metaplasia. For example, when you are seeing a columnar to squamous transformation, see what I am going to show you here. When you are seeing a columnar to squamous metaplasia, squamous cell, right? Columnar to squamous cell transformation. So this will be considered as example of squamous metaplasia. So end outcome is squamous cell. So this will be called as squamous metaplasia of the respiratory tract. So this will be called as squamous metaplasia of the respiratory tract. Squamous metaplasia. So squamous metaplasia. So now you can see whatever is the end outcome, nomenclature will be based on those things. So now we can easily make it out, right? Okay, columnar cell has been replaced by squamous cell because of the smoking in respiratory tract. And this is called as squamous metaplasia, and squamous metaplasia is the most common type of metaplasia in human body. So that is the important future question which examiner can ask you, right? So squamous metaplasia is the most common type of metaplasia in human body. So that is the important final question. Squamous metaplasia in respiratory tract, right? This was the AIMS question in 2013. Squamous metaplasia is because of which vitamin deficiency? So here we have to understand squamous metaplasia in respiratory tract is due to vitamin A deficiency. So that was the question in AIMS exam. So what I want to tell you, in future exam, examiner can ask you, squamous metaplasia is due to vitamin A deficiency or vitamin A excess. So when you will go through the Robbins, you will find that it is vitamin A deficiency also, and it is because of vitamin A excess also. Both can induce squamous metaplasia. So just remember this point for future exam point of view. Vitamin A excess can also induce squamous metaplasia. So answer will be both of the above. Squamous metaplasia of respiratory tract is because of vitamin A deficiency, vitamin A excess, or both of the above. Answer should be both of the above. Both vitamin A deficiency and excess can induce squamous metaplasia, right? So this is example of squamous metaplasia of the respiratory tract. There is another important metaplasia, which is the pathological metaplasia, which we remember, which we see often in our clinics also. That is the Barrett's esophagus. So now the second one is the Barrett's esophagus. Second example of the pathologic metaplasia is the Barrett's esophagus. What is Barrett's esophagus, right? What is Barrett's esophagus? Barrett's esophagus, what is the most common cause? Why there is a Barrett's esophagus? Because of the gastroesophageal reflux. When the gastric content is high, pressure will be more, it will be having reflux, it will come up. And when gastric content will come up, what is gastric content? Acidic or alkaline? Acidic. Acidic content, when they are coming up into the esophagus, lower part of the esophagus, they will cause damage or they will cause change or metaplasia, right? So that is called Barrett's esophagus. So what I wanted to tell you, most common cause of Barrett's esophagus, most common cause of Barrett's esophagus, when examiner had asked this question in All India DNB exam, your answer must be gastroesophageal reflux disease. Gastroesophageal reflux disease, right? So gastroesophageal reflux disease is the most common cause of Barrett's esophagus, right? What happens in this gastroesophageal reflux disease? Already I have told you, there will be reflux of acidic content from the stomach to the esophagus, lower 1/3 of the esophagus, right? So because of this reflux in the esophagus, what you will notice here, when you see biopsy of the esophagus, right? Esophageal biopsy, when you will notice normal esophagus, they will be having flat and squarish squamous cells. So all these are squamous cells, squarish squamous cells. So squamous cells will be flat or squarish. In biopsy specimen, they may be elongated also, because these are tissue and they are amenable to stretch or they are prone to stretch. So when this squarish thing will be stretched, they may be elongated like this also in your biopsy specimen, right? So keep that in mind. Either they will be squarish or elongated in case of biopsy. So this is the normal squamous cell within the esophagus, right? Because of the gastroesophageal reflux, disease, as I said, there will be stimulation given to the squamous cell. This squamous cell, mature cell, will be sending signal to the stem cell. Now stem cell will be having reprogramming of the stem cell, and after reprogramming of the stem cell, they will form the new kind of cells which can sustain this gastroesophageal reflux of the acidic content of the stomach. Acidic content is coming up, they are putting stress on the squamous cell, signal has been sent to the stem cell will be going for reprogramming. Under this reprogramming, what they are going to do, they will form another type of mature cell. So now they will stop squamous, they will form the new type of cells which will be looking like a column, like this, like a column. So these are the tall columnar cells. So this is how a columnar cell will be formed, and this is called columnar cell, and that is why this metaplasia is known as columnar metaplasia. If you remember, earlier I have told you, metaplasia name is given on the basis of end outcome of the cell. So what is Barrett's esophagus? That was the question in DNB and All India exam. What is Barrett's esophagus? It's a columnar metaplasia of esophagus. Remember this point. Barrett's esophagus is the columnar metaplasia of the esophagus, right? What happens in this columnar metaplasia? If you look at this cell, they will be having nucleus. And when you will look at the columnar cells of this Barrett's esophagus, you will notice there are certain vacuolated structures, or you can see there are vacuoles, and within these vacuoles you will notice there are certain substances which are called as mucin. They are having mucin, right? So what I am showing you here, this is mucin within the vacuole. So mucin is present inside this vacuole, and this mucin containing vacuolated cells are called as goblet cells, right? So all these cells are called as goblet cells. So this mucin containing cells are called as goblet cells. So goblet cells. Now the question was in DNB exam, will you find goblet cells normally in an esophagus? Will you find goblet cell in a normal esophagus? Answer is no. No goblet cells will be seen in normal esophagus, right? So whenever you find goblet cell in an esophagus, this may be a sign of metaplasia. So where you will find goblet cells normally? Where you will find goblet cells normally? In intestine. Where? In intestine. That is why when you find the goblet cell in esophagus, this is called as intestinal metaplasia. So remember, because of this only, this is also known as intestinal metaplasia. So that is why it is called as intestinal metaplasia, because goblet cells are usually or normally seen in intestine, and now we are seeing them in esophagus. So that is why it is called as intestinal metaplasia. And always remember, this is an important MCQ. Intestinal metaplasia is hallmark of Barrett's esophagus. Hallmark of Barrett's esophagus. So intestinal metaplasia is hallmark of Barrett's esophagus. Another important and frequently asked question in exam. Right, so this is another important and frequently asked question in exam. Hallmark of Barrett's esophagus is intestinal metaplasia. What does that mean? Goblet cells are seen. So the question comes to our mind, how we are going to discriminate this goblet cell of the Barrett's esophagus from the normal goblet cells of the intestine? That was the AIMS question. That was the AIMS question previous year. How we are going to discriminate this goblet cell from the intestinal goblet cells? So remember, as you understand, when I started the pathogenesis of metaplasia, what I said, most common cause is the gastroesophageal reflux disease. Reflux of what? What? Stomach. Stomach food. What is stomach content? Acidic or alkaline? Acidic. And that is the catch point. Acidic content. That is why whenever you are going to use one stain, what is that stain? We are going to use one stain for goblet cells which is containing mucin, right? So what I am going to show you here, mucin stain. What is the mucin stain? This was All India and AIMS question. Mucin stain is Alcian blue stain. So Alcian blue stain is used for mucin. Alcian blue stain is used for mucin. That was the question in AIMS exam. So Alcian blue stain is used for mucin staining, right? So Alcian blue stain is used for staining of the mucin within the goblet cells. So how we are going to discriminate this goblet cell is of intestinal origin or if it is residing into the esophagus? To discriminate that, we require a specific pH, and that is the catch point for that some simple question. Alcian blue stain pH must be, Alcian blue stain pH must be, you tell me what your logic says. It should be alkaline or acidic. See this, what I said in the pathogenesis, try to correlate that. Stomach content is coming up which was acidic. So what kind of pH we should be requiring for Alcian blue? Acidic. pH will be acidic. So what is acidic pH? It will be 2.5. So acidic pH, this is the important MCQ. Acidic pH is important, right? So acidic pH is required for goblet cells of the Barrett's esophagus. Whereas alkaline pH will be used for, alkaline pH containing Alcian blue will be used for the intestinal goblet cells. This is very, very important to understand, and that is why examiner asks question from here. Right? So we were discussing about Barrett's esophagus where columnar cells were replacing the squamous cells, and that is why it is called as columnar metaplasia, because of the presence of the goblet cells which is usually not present in normal esophagus, is called as intestinal metaplasia. And whenever you find intestinal metaplasia, it's hallmark of Barrett's esophagus. How we are going to change, how we are going to discriminate this is the intestinal goblet cells or esophageal goblet cells? By using one special stain. AIMS question, Alcian blue stain. what should be the pH? Acidic pH, because gastric content is acidic, right? So there will be pH 2.5. So acidic pH 2.5 will be required to obtain goblet cells in our Barrett's esophagus. After seeing all these things by a pathologist, what a pathologist will think about? Means why a pathologist is worried about it? Why a surgeon who has sent this gastroesophageal biopsy is worried? Why? What is the reason? Reason for worry is the risk of adenocarcinoma. So remember, what is the Barrett's esophagus risk? So remember this point, Barrett's esophagus, what is the risk? Risk will be adenocarcinoma of esophagus. Adenocarcinoma of esophagus, right? So that is the another important MCQ which examiner had asked. Adenocarcinoma of esophagus can be because of Barrett's esophagus, right? So this is the important thing. We have to keep that question in our mind. And again, I will tell you one more important thing. What is the significance of the metaplasia? Usually, see my wording, see my language, because that is the thing we have to focus on. Because in exam, examiner will ask question on these kind of exceptions, right, which I am going to explain you. What I am going to explain you here, if you remember, when we discuss what is metaplasia, what is the danger? Metaplasia is dangerous because they are having risk for cancer transformation, right? So what is that root? Metaplasia will be undergoing to dysplasia, and then dysplasia will be going to carcinoma, right? So that is the important thing we have to understand. Metaplasia, dysplasia, carcinoma. So that is what it has been given in our standard textbooks also. Okay, metaplasia is a swell for carcinogenesis. Is it really true for all the cancers? Or is it really true for all the conditions? Is it really true for all the conditions? Or is there any exception? So what I wanted to tell you, that metaplasia, dysplasia and carcinoma, this sequence, they are having one notable exception. They are having one notable exception. And what is that exception? We have to keep that in our mind. Apocrine metaplasia of the breast will have no risk for malignant transformation. So this point we have to remember. What is that exception? That exception is apocrine metaplasia of breast tissue, right? So apocrine metaplasia of the breast tissue is having no risk for malignant transformation. This point we have to keep in mind for our future exam. So apocrine metaplasia of breast tissue is having no risk of malignancy. No risk of malignancy. So this is one example which we have to keep in mind for the future exam, right? Usually we say that metaplasia, dysplasia and cancer will happen, but remember, it is not always true. There is one exception, and that is apocrine metaplasia of the breast. There will be no risk of malignant transformation, right? So these are important points regarding cellular adaptations, in which we have seen altered physiologic stimuli, or because of non-lethal injury, all these changes are happening. And when you will remove the stimuli, they will come back to the normal state, right? They will come back to the normal state. And here, before we move to the next topic, I would like to show you this image of Barrett's esophagus, right? This image of Barrett's esophagus. So here, before I start, I would like to tell you one more thing, which is applicable for all the histopathological images as well as hematological images in pathology. Whenever you see a question in your exam, which is the from the pathology, always remember three important things. Number one is rule of 5. Rule of 5 means you should always try to see 5 areas in this slide. 1, 2, 3, 4, 5, right? So there are 5 areas in the slide. Whenever you are appearing for your exam, seeing any histopathological specimen, or whenever you are studying, doing self-study also, this is called rule of 5. All 4 corners and the center. All 4 corners and the center. All 4 corners and the center. So rule of 5, number 1. Then rule of observation. Then observe what kind of cells are here, what kind of cells are here, right? So number 2 is the rule of observation. Number 3, after observation, as a good clinician, what we expect from us? After observation, comparison. Compare the things. Ki how is this? Comparison is how is this? So compare how is this cell here, how is this cell here, and how is the condition of cell here, right? So these are the 3 very important rules to understand any image given in our pathology textbooks, right? So rule of 5, rule of observation, rule of comparison. These are the 3 very, very important things we have to follow in our entire pathology specimen or entire pathology image information understanding, right? So the same thing I am going to show you here. This is a case of an 33-year-old engineer who was working in a multinational company. His food habits were deranged. He was suffering from gastroesophageal reflux disease. Esophageal biopsy was done, and on esophageal biopsy done, this is the esophageal biopsy. So what is your inference? So when you are seeing the picture, so as I said, rule of 5, rule of observation, rule of comparison should be the thing which should be running in your mind to understand any image given in your exam. So when you are seeing 1, 2, 3, 4, 5 areas and we are understanding these things, can you see the cells here? These are squarish, or you can see the soft tissue, they are elongated also. So what are these cells? These are the squamous cells. These are the squamous cells of the esophagus. Esophageal biopsy, so we are looking at the squamous cells of the esophagus. But when you are coming down to the gastroesophageal junction here, right? So you can see squamous cells are here also, squamous cells are here also. But when you are going to the junction lower down, so lower part of esophagus, you can notice in this area, which I had marked in a square. In this area, you can see there are cells which are elongated, columnar, right? So these are columnar cells, right? So tall columnar cells, and you can see the nuclei at the base, and you can notice there are vacuoles which is containing mucin. So these are mucin containing goblet cells, right? These are mucin containing goblet cells. So these are all columnar cells. So we can notice that these are columnar cells, right? So in a column, and these are columnar cells. That is why it is a columnar metaplasia, right? Columnar cells are tall columnar cells, and they are having mucin containing goblet cells, right? So that is why it is called as intestinal metaplasia. So when you are observing and comparing, you can see that how squamous cells are getting transformed into columnar cells at the gastroesophageal junction. So this is the Barrett's esophagus. And whenever we are coming across with this kind of biopsy, what will be the thought process going in a pathologist's mind? It's a Barrett's esophagus, and it can be a future risk for the adenocarcinoma of esophagus, right? Now the question comes to our mind, that this goblet cell which we are looking at here, how we are going to identify this is the goblet cell? To identify this goblet cells, we need to have a special stain, Alcian blue stain, and in Alcian blue stain, this goblet mucin will be turning out to be blue. As you can see, the name is Alcian blue. So Alcian blue stain, goblet cells will be standing out as a blue. So now you can see this is the picture of Alcian blue stain, and here you can notice all these are blue color mucin, right? So this is the Alcian blue stain, Alcian blue stain. And if you remember, Alcian blue stain, as I said, mucin will be blue. So Alcian blue stain is used for mucin. And what will be color of mucin? Mucin will be blue. What will be the situation of this Alcian blue stain? pH will be acidic. pH will be 2.5. So that is how Alcian blue will detect these goblet cells of the esophagus in Barrett's esophagus, right? And this diagram and this point has been a question of previous year AIIMS exam, right? So this is all about cellular adaptation. Now, when cell will have lethal stimuli, then they will transform into cell injury, which we will discuss in next topic called as cell injury.